Now after fertilization, the next phase is post-fertilization phase, which is marked by formation of endosperm and embryo. Endosperm is nothing but this primary endosperm nucleus. It goes into divisions and give rise to free nuclei. Large nuclei are formed. Now once these free nuclei are formed, cell wall formation starts, which gives rise to endosperm. Now in certain cases, endosperm is completely consumed in the formation of embryo itself, and in certain cases endosperm is still present and further helps in germination of the seed. The example in where endosperm is completely consumed is groundnut, pea, and where it is present is coconut. You can see when we break down the coconut, there is water as well as white colored kernel. This water is nothing but free nuclei endosperm, and the white colored kernel is nothing but endosperm. Endosperm is nothing but a food reservoir. It is having very thick cytoplasm. It consists of food reserve, which is useful for the development of embryo, or which is useful for the development or the germination of the seed. Now embryo is made up at or starts forming at the micropylar end of embryo sac. This development of embryo at the micropylar end of embryo sac is known as embryogeny. Embryogeny is similar in cases of both monocotyledons as well as dicotyledons. This is a dicot embryo. This is plumule. Here is epicotyle. These are two cotyledons. This is radicle and this is root cap. In case of monocotyledons, only one cotyledon is present, and in case of grass, these cotyledons are known as scutellum, and further this root cap is not there. There is thick sheath of cells which is known as coelurhiza. Now the zygote gives rise to proembryo. This proembryo gives rise to globular embryo, which gives rise to heart-shaped embryo, and further it gives us the mature embryo. So this is the post-fertilization event. Endosperm as well as embryo, these two are formed.